Dear students, this is Dr. Pooja Thakkar, a biology expert. We are in the first session of Biomolecules, and in today's session we'll discuss regarding carbohydrates. Okay, first I would like to give you the detailed introduction of biomacromolecules. Now, what do you mean by biomacromolecules? Biomolecules, as the name suggests, the molecules which are of biological origin, but they are very big in size, so they'll be considered as biomacromolecules. Each and every living system contains various types of biological molecules, or simply biomolecules, which are basically serving as the building blocks of the life, and these macromolecules enable the living system to perform vast array of functions. Now, the biomolecules include the chemicals which are composed of mainly carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. So these are the basic constituent component. Okay. Now, biomolecules include the small metabolites, or they can be called micromolecules, or they'll also contain the bigger sized molecule called macromolecules, as we discussed. So what these basically biomacromolecules are, from the example you will be able to understand the concept more clearly. So these are the four biomacromolecules, that is carbohydrates, lipids, proteins, and nucleic acid. So first we'll discuss regarding carbohydrates. Carbohydrates are basically commonly called as sugars. These are the organic molecules which are made up of carbon, hydrogen, and oxygen. As we are already knowing the structural, or the chemical composition of glucose or sugar, that is C6H12O6. So they are having the carbon, hydrogen, and oxygen, specifically with the hydrogen to oxygen ratio, that is 2:1. In C6H12O6 also you will be able to notice hydrogen are 12, whereas oxygen are half of 12, that is 6 in numbers. Okay, so every time the carbohydrates are maintaining hydrogen to oxygen ratio, that is 2:1. Now, as we discussed, the monosaccharide, that is glucose, it is having 6 carbons. As you can see in the diagram, they are having the orientation of different components on them, that is the inorganic molecules on them, that is hydrogen as well as the hydroxyl group. But here we are seeing the aldehyde group at the top, okay, which is CHO. Sometimes these kinds of carbohydrate molecules can also bear ketone group, so that kind of sugar is called keto sugar, and this kind of sugars which are having aldehyde group in them, they are called aldo sugar or aldose sugars. Okay, so we'll discuss that in detail, that each sugar molecule have a free aldehyde or a ketone group. Based on that, the carbohydrates are classified into two categories based upon the type of functional group they are carrying. Okay, either it is aldose or ketose. So these kinds of sugars are called the reducing agents, which are having free aldehyde group on them. Okay, now in the next slide, what we need to discuss, that how this carbohydrates are basically made from carbon, hydrogen, oxygen, as well as the type of them. Okay, so for that, to understand the basic detail regarding varieties of disaccharides or the polysaccharides, first we need to undergo the analysis of monosaccharides, that is the monomer unit or the basic sugar unit, okay, of the carbs. That is the simplest subunit of the carbohydrate is called monosaccharide, or in short monomer, which is basically made up of 3 to 7 numbers of carbon atom. As we have discussed here, the glucose is having 6 carbons. Okay, so that is the monomer. Okay, so each kind of monosaccharides are basically having 3 to 7 carbon atoms. These cannot be further hydrolyzed into simpler chemical compound. The hydrolysis of these small kinds of monosaccharides into individual components is not at all possible. Okay, now what we need to understand is what are the properties of these monosaccharides. Now, how you will identify either it is monosaccharide or something else. So monosaccharides are sweet, They are crystalline as well as they are water soluble. The best example is glucose, as we have discussed, which is aldohexose. The second example is fructose, which is a ketohexose, which is mainly present in fruits, so it is also called as fruit sugar. Okay, so these two are the best example of monosaccharides, that is glucose and fructose. Now what we need to discuss is when the two sugar molecules or the two monosaccharides are joining together, they are forming the disaccharides by the help of a bond called glycosidic bond. You will be able to notice varieties of disaccharide in this particular diagram. The first one is called sucrose, which is made up of a glucose and a fructose. They are linked together by forming a bond in between O is present. So these kind of bonds are called glycosidic bond. Now the next example is lactose, which is made up of a galactose and glucose. Likewise, the next example is maltose, which is constituting two glucose unit together. Okay, so these way, by forming glycosidic linkage, either it is alpha 1,4 glycosidic linkage or beta 1,4 glycosidic linkage, or it can also form 1,6 glycosidic linkage. The type of you can say the components which are involved in bond formation, as per that the 1,4 or 1,6 kind of linkage can be given. Okay, so disaccharides are basically of varieties of kinds. Sucrose is also called as table sugar. Lactose is also called as milk sugar, as well as maltose is also called as malt sugar. Okay, now the next category is of oligosaccharide. Now what do you mean by oligo? Oligo is a small group or a small chain, you can say. Okay, so when a small number of monosaccharide units are joined together, they are called oligosaccharide. These are involved in the cell recognition and cell binding process. This kind of oligosaccharides we have studied during the plasma membrane. Whenever we are discussing regarding glycoprotein or glycolipid, you can go onto those on that particular video to understand it more clearly, that the lipid constituent of the plasma membrane as well as certain kinds of proteins are carrying few sugar moieties on them on a chain based structure. Okay, so those kind of structures are basically called an oligosaccharide, which is a shorter chain of glucose. Okay, and they are doing the glycosylation process. Why they are doing glycosylation? They are adding the sugar moieties on lipids as well as proteins. So those kinds of lipids and proteins are then called as glycoproteins or glycolipids. We have also discussed the roles of these glycoproteins and glycolipids during the time of plasma membranes detail. Okay, so those are basically involved in the cell recognition. Based upon the type of sugar which is present on the boundary of plasma membrane, you can identify particular kind of cell, as well as they are having the sticky nature, so they'll help the cell to bind with the other cell as well as to the other substratum. Okay, so this is all about oligosaccharides, and these are the structures of disaccharide. Now we are moving on to the polysaccharides. When many monosaccharide units are joining together, they form a long chain or a polymer called polysaccharide. These are a complex carbohydrate molecule which are having exactly different properties than the monosaccharides. You can say they are not at all sweet, they are not at all crystalline like monosaccharide, as well as they are completely insoluble in water. Okay, now what are the examples? The first and the most important example is cellulose. So cellulose is a homopolymer. Whatever we are discussing here, whatever examples we are discussing here, all are homopolymers. Now what do you mean by homopolymer? The polymer which is made up of a same kinds of unit. All the monosaccharides which are involved in polymer formation are same basically. Okay, so each and every monomer here is the glucose. Okay, so cellulose is the homopolymer of glucose, and it is found mainly in the plant cell wall, and they are forming the plant building material. The next example is starch. It is present sorry, it is basically made up of a same kind of monosaccharide unit called glucose, but it is found inside the plant cell as a food reserve material. Starch is the byproduct, not a byproduct, it is the main product of photosynthesis. Okay, so whenever a plant cell is involving, or typically a mesophilic cell is involving in the process called photosynthesis, by the utilization of carbon dioxide as well as water, they are creating glucose units together, and the combined structure of glucose is called starch, which is a reserve material in plants. The third and the most important example is glycogen. So glycogen is also a homopolymer made up of the same monomeric units called glucose, and they are found in the animal cell as the food reserve. Likewise, in the plant, we are also having the food storage component or the food reserve material in the format of glycogen. And the last example is the chitin, which is also a homopolymer, which is made up of N-acetyl glucosamine. This we have discussed during the gram staining process, that a bacterial surface is typically involved. Bacterial surface is mainly carrying these kinds of unit, N-acetyl glucosamine and N-acetylmuramic acid, for the formation of peptidoglycan layer. So here also a homopolymer called N-acetyl glucosamine, that is a variant of glucose only, the glucose unit with a one amino group in them, as well as the acetylation of that particular component take place to form these kind of component. Okay, that is N-acetyl glucosamine. They are binded together to form a long chain called chitin, and which is the most common structure commonly found in the fungal cell wall as well as the exoskeleton, that is the outermost skeleton of certain insects. Now, based on the location where these kinds of polysaccharides are actually present, you will be able to get the idea that what is basically the biological function of those carbohydrates are. Okay, so the first important function is they are mainly serving as the energy sources for the living organism. For example, you can say a starch or a basic unit glucose. They are serving as the reserve storage for the living organism, starch in case of plants as well as glycogen in case of animals, you can say. And they are playing a structural role, okay, to form or to create varieties of membranes. So cellulose in plant, you can say, and chitin in case of fungus, as well as exoskeleton of insects. Okay, so this is the function of carbohydrate, and where the storage exactly takes place, as we have discussed, in the form of starch, they are stored in the plant, as well as in the form of glycogen, they are stored in the animals. Okay, so this is the very basic information regarding carbohydrates. You can put your queries in the comment box below, and please do like the video and subscribe the channel. You can also call or message on the number, that is 9909804647, for more information or your queries. Thank you so much.